To introduce our speaker, he's Professor James DeGregori. He holds an endowed chair in lung cancer research at the University of Colorado. James received his PhD from MIT, and he has an undergraduate degree from UT Austin. James and his lab investigate cancer evolution with an eye towards advancing therapy, and including through an evolution-based model that they call adaptive oncogenesis. And by the way, James has a book of that same name published by Harvard University Press. I'm going to paste in here a link to that book for those of you who are interested. Okay, with that, I'm going to turn things over to James. Take it away. Thanks for joining us.
Great. Thanks so much, Charlie. And thanks a lot, Meredith, for organizing this. And I want to thank Randy Nessie for having invited me as well. It's really a pleasure to do this. And I'm going to go ahead and share my screen. So I will try to keep this to 20 minutes, and then we can open it up for discussion, which I think is going to be best. By the way, when we do the discussion, it's really great if people turn on their camera. That way, during the pandemic, we're already separated, but at least we could see each other's faces, during questions and answers and stuff like that. So anyway, I'll be talking about how recent characterizations of somatic mutations in humans inform an evolutionary understanding of aging and cancer. So I hope everyone recognizes that 90% of cancers develop after the age of 50. So cancer is largely a disease of the aging, not exclusively, but largely. And so this is actually SEER data for all cancers combined, including leukemias. And you can see that pretty much you don't want to pass your 40th, 50th birthday because everything bad starts happening to you. So really the question is why? Why is this happening? So from the traditional perspective, if you read your cancer biology textbooks, or if you read most of the literature out there, this is really due to multiple factors. And hold on, I'm going to change my pointer to something a little more visible, including time, cell divisions that happen in our body, sort of endogenous processes, extrinsic factors like exposures through cigarette smoke and stuff, plus our inherited genetics. And the idea is this really all just comes down to mutations, and that mutations really are the drivers of cancer. But I think we should step back and ask, well, how has evolution been, what has led to the diversity of species that now inhabit our planet? And, we know that species have changed dramatically over the hundreds of millions of years, certainly since, animals first developed. And this has really been driven by massive changes in environments, either due to extrinsic factors like meteors, intrinsic factors like volcanic eruptions, huge changes in the gases, in the atmosphere, changes in temperature, and so forth. And it's really been environmental changes that, for example, have driven most of these transitions. So environmental changes drives, the selection for new types that are adaptive to the new environment. And this is what drives change. So why should we think of cancer as anything different? So what I'll argue is that our understanding of cancer development and risk needs to incorporate how tissue environmental changes drive selection for oncogenic events. I'll also mention that all artwork that I show was done by my son, Michael, and he actually did all the artwork for my book as well. So we've done a lot of different we've taken a lot of different angles to argue for the importance of microenvironmental change in driving cancer evolution. But I'm just going to show you one computational study done by a fellow at my lab, Andrew Rosak. And what he did is he just took SEER data, and you can see this is SEER data for different leukemias, different solid cancers. And the important thing is these numbers in parentheses are the numbers of drivers mutations that have been estimated for these cancers. And they're between 4 and 11 for colorectal, down to like 1 for mesothelioma. And they also range from about 1 for CML up to several different mutations for AML and CLL. But what I find really striking is if you just normalize all these to their median, to their maximums and their minimums, the graphs fall right on top of each other. In other words, the shape of the graph, even if the absolute incidence is different, the shape of the graphs are very strikingly similar. And even a similar graph is seen for clonal metapoiesis, which is known to be driven by single driver mutations that happen in hematopoietic stem and progenitor cells and that lead to expansion of clones as we get old. And this is even more amazing if you consider that these different leukemias and different solid cancers initiate and stem cell pools with very different organizations. And, with different numbers too, there's estimates that there's at least 10 million different stem cells that maintain our colon, and about a couple 100,000 stem cells that maintain our hematopoietic system with completely different organizations. So to me, this is striking, and this suggests that one cannot simply model this as the number of mutations required. So Andre developed a Monte Carlo model, and I won't go into the details of the model, but it's in eLife. And what he showed is the only way to really reconcile all these different factors into these common incidence curves was to have differential selection through life, meaning that mutations that change phenotype tend to be selected against in youth. Basically favor the status quo, while these same mutations can be positively selected, or at least some of them late in life. And it's this differential selection that allowed us to basically model these different cancer incidences despite the different numbers of driver mutations, despite differences in stem cell pool and so forth. So essentially what we're arguing is that we need to look at this from a higher perspective, and that these common curves are really the result of natural selection. And for someone who thinks evolutionarily, this really shouldn't be a surprise, in that there's selection against cancers that will reduce our fitness at least through about half of a century. And in some ways, I find that quite amazing. It's amazing that you can have a body of a human with 40 trillion or so cells, and then we have such low cancer incidence, at least through reproductive years. So really, this comes down to understanding that for most of our evolutionary history, the number of individuals that survived past, let's say, 60 was relatively small. It wasn't nil. There were some individuals that made it to old age, but you can imagine that there'd be minimal selection against cancer and other diseases of aging beyond the age where most individuals would already be dead by other causes, and certainly beyond the ages where they were likely to pass their genes on. And there's obviously confounding factors here. We know that, older males in some tribal societies actually have increased, I should say, access, I don't know if that's the right, to younger females and so forth, but so it could have been that it wasn't like your fitness was nil when you're out here, but still your odds of reproducing and passing those genes on successfully was less. And if you look at modern incidence curves for different diseases, you can see that they're largely occurring in what would have been post-reproductive years. So what we would argue is that, and I mean, this is based on classic evolutionary theory going back to, Williams and Medawar and others, that there was an investment in our maintenance through the years when there was most likely to be a payoff from that investment. And this is one of the reasons that I think that we maintain our tissues quite well for half of a century. And then we don't just go downhill, but we pick up speed as we go down. Our tissue decline is not continuous through life. It is largely maintained and then declines late in life. So then what does this investment in tissue maintenance during youth and the waning of this investment in old age have to do with cancer? And this is the theory that Charlie referred to, adaptive oncogenesis, that my lab's been developing. And so this theory really has two critical components. The first one is essentially what I actually find to be the more amazing fact is, how do we not get cancer largely through half of a century of life? And what we argue is this isn't simply by avoiding mutations. And I think the two journal articles I've chosen help demonstrate this, that mutations are going to happen. But if somatic selection opposes the selection for these mutations, at least mutations that could be damaging, then it's going to maintain the status quo. So this is no different than basically the stabilizing selection that we all learn about in, on evolutionary biology 101. That if you have a population of animals, bacteria, whatever, and that they've evolved to be at some peak of fitness because they're in a constant environment, then change will tend to be basically reduced fitness, because in any direction, phenotypic change is going to be selected against. And so even if there was another phenotype, in this case, this could be a malignant genotype if we switch back to talking about somatic cells, with a higher fitness, if getting there meant going through some fitness valley, it's going to be unlikely to happen. And I'm using words like that on purpose, because it's not like a healthy 30-year-old can't get cancer, just he or she is very unlikely to get cancer. So this might still happen, even if it's very rare. And what this requires investment in the tissue environment. Because what we argue is that the wild type genotype of a stem cell, for example, is well adapted to the natural tissue niche, the youthful tissue niche. So the stem cells in your lung are adapted to a healthy young lung. They're not adapted to the lung of an older person. And so that becomes the problem, is that we don't stay young. We don't invest in the tissue maintenance forever. We, meaning natural selection, hasn't. And so at some point, our tissues do go downhill. And of course, then we also, have activities like smoking that makes this decline even more precipitous and more dramatic. And so whether due to extrinsic factors like our environmental exposures like through smoke, or intrinsic factors like aging, we know that this relationship between the stem cell genotype and its environment becomes degraded. And the stem cell basically finds itself in an environment that is no longer adapted to. And so then you're going to get selection for within these stem cell pools for new phenotypes that might be adaptive to this environment. And sometimes these phenotypes can be malignant. And this is an important point. I would say most of the time they're not malignant. And we'll see this in some of these papers, but sometimes they can be malignant. And this increases the odds of cancer. So essentially, if you think about this, what we're basically arguing is that we may accumulate genetic damage, genetic meaning including epigenetic damage in our cells, somatic cells, throughout our lifetime. But this is largely acted on by stabilizing selection, at least through years of likely reproduction for most of our evolutionary history. But late in life, as our tissues decline, and which can, of course, be accentuated by activities like smoking and other things, you can now get positive selection for the same phenotypes that were negatively selected before. And it's this change, this switch, that can lead to this increase in selection for certain cancer phenotypes. So is there evidence for this? And actually, my group has accumulated quite a bit of evidence. I'm just going to show you one slide of such evidence. And this is work from a postdoctoral fellow who was in my lab, Curtis Henry. He's now an assistant professor in Emory. And what Curtis did is he wanted to ask, well, how does old age influence the selection for particular oncogenic events? And he would take cells from a young mouse, all the same cells, and a small fraction of them would express an oncogenic event. And he put these into recipient mice, and he did it in a way that was not inflammatory. And I won't have time to go into the details of that, but these could be either young or old, or they were old mice that expressed this transgene that reduced inflammation. And what he finds is in the young mice, just as the theory predicted, because we use this non-disruptive method, he showed in the young mice, these oncogenic events were not advantageous. And this really flies in the face of conventional wisdom, where everyone thinks the oncogenic mutations sort of inherently confer an advantage to the cell, but they don't. But in the old background, we do get this advantage that gets conferred. And you see this expansion of the clones. But if we blocked inflammation, if we just changed one thing about the environment of these old mice, we could largely prevent that expansion. And just to give you one example of how we see this at the sort of molecular level, we see that the old progenitor cells have reduced signaling, and they also have reduced expression of key genes important for DNA replication. So the old cells aren't doing as well. They have reduced fitness within their microenvironment because of this. And we know this is largely due to the microenvironment, because in the blue bars, these are the same cells in the old mice that have the anti-inflammatory transgene. And we can largely prevent this from happening. But if you put in the RAS oncogene, you can see that you can restore these signaling and gene expression parameters, explaining why it's adaptive in the old environment. But in the young environment, they're already what we would call at the peak levels. So RAS is not advantageous. And the same is probably true for the cells that express the anti-inflammatory transgene. We've taken away that selective advantage by maintaining the fitness of the cells. So there's a lot of recent examples in humans of context-dependent selection, particularly where we see increased selection as people get older, also during inflammation, which I'll show you quickly. So I'm just going to show a couple figures from the papers the assigned readings. And so this is the first one from Inigo Martin Cordenas and Peter Campbell's group at the Sanger Institute. And what they did is they just sequenced these little small pieces of esophageal epithelium. And they sequenced small enough pieces that they could basically see somatic mutations. And what they find is they get this incredibly common occurrence of Notch1 mutations, plus some other mutations that are found within this epithelium. And using a sort of a modified DNDS algorithm, they could show there's really strong positive selection. I don't know if you're all familiar with DNDS, but basically anything above 1 is positive selection. And typically in evolutionary biology, selection much above 1, even 1.1 or 1.2 is considered quite good. They're getting DNDS ratios of up to 150, which is just really strong positive selection. And when they look at the distribution of the mutations, they can see that they're very much the same sorts of mutations that you would get in the cancers. So they seem to be cancer type mutations. But the other cool thing is that at least for the Notch1 mutations, they occupy a very large fraction of the epithelium. In fact, if you look across the different ages, by the time you're 50, about half of your esophageal epithelium, whether or not you smoke, I'll note, or whether or not you drink, is basically occupied by Notch1 mutations thought to be tumor suppressive, thought to be, since Notch1's a tumor suppressor, thought that these should be pro-cancer mutations. And yet right now, half of my epithelium in my esophagus is Notch1 mutant. But what you notice is that while a large fraction of the epithelium of a typical person is Notch1 mutant, actually that far outpaces the frequency of these Notch1 mutations in the cancers associated with the same tissue. So essentially, most of the cancers are coming from the part of the epithelium that does not have the Notch mutations, which I find really interesting. So, and that's just, I wanted to point that out. In fact, I'll just make one point that the authors pointed out, and they estimate there are about almost 4,000 positively selected driver mutations and about 17 centimeters squared of normal esophageal epithelium sequence. That number is comparable to the yield of driver mutations obtained from sequencing more than 1,000 cancer genomes. The bottom line is we are absolutely loaded with what we used to think of as oncogenic mutations. I would say we're currently doing estimates, but I put the estimate as somewhere close to 1 trillion oncogenically mutated cells in our body by the time we're middle-aged. And despite this high frequency, should we all freak out? And I would say, of course not, because we already know what cancer incidence is, and knowing that you're loaded with mutations doesn't change your risk of that. But the other thing I'll point out is that, so we actually, from this paper, it made us think that, well, maybe this is telling us something. Maybe this Notch peak, this Notch mutation is indeed an adaptive mutation. It's clearly adaptive. I mean, the DNDS values are above 100, but that as we age, that you can get adaptation up this peak. But what this does, even though there could be potentially other more malignant routes for somatic evolution, that these other malignant routes basically become less favored when there's this other one. Essentially, I would call it a decoy fitness peak. And so these Notch mutant clones may in fact be tumor suppressive by allowing a less malignant alternative to a more malignant peak. And at least they can function until we get old enough. And so if you look at when the p53 mutations tend to rise, they tend to rise very late in life, or at least expanding out. I'll mention that these mutations are certainly occurring throughout your life. It's just they're being selected for late in life. And so natural selection only needed to protect us, at least maybe through these middle-age periods where we still had some value in terms of contributing to reproductive success. So in the second paper, which I'm about done with my 20 minutes, and I'm only going to show 2 figures of this. This is looking at something different. This isn't looking at aging-dependent selection. They looked in the guts of people with ulcerative colitis, and they basically see very frequent mutations in this IL-17 pathway that inactivate the pathway. Essentially, people with ulcerative colitis have this chronic inflammatory disease, and what they're basically showing is that you frequently inactivate mutations that would normally be downstream of this inflammation. So essentially, they're becoming inflammation resistant in some respects. And I just want to point out this one factor again. So many of these mutations, like in this NFK-BIS gene, which is in this pathway, they're very common in people with ulcerative colitis. In fact, they can represent huge swaths of the colon. And yet they're unbelievably uncommon in people when they get the cancer. So again, I would argue that these, if anything, are protective because they are very unlikely to contribute to the actual cancers. So I used to think that natural selection had acted to basically limit all somatic evolution, and that was wrong. So in fact, I probably even say that in my book, but just note that that is not correct. And of course, I wasn't referring to things like B cell and T cell evolution within our bodies, because clearly that's good somatic evolution. But I would now argue that natural selection has just acted to limit the somatic evolution that reduces our fitness. And there's a lot of somatic evolution that happens within us that may actually be helpful to being tumor suppression. So we may not be able to prevent most aging-associated mutations, but the question is, can we modulate their impact by manipulating the microenvironment, maybe to better favor, for example, these decoy type beats, the Notch1 mutations, the NFK BIS mutations, in other words, favor the more benign type routes. And that is it. And I would love to take questions. And again, please turn on your camera when you ask a question, or get discussion going. Let's, and so I don't have to be the only one answering questions. If someone else wants to contribute, please jump in.
James, that was great. Thank you very much.
Thank you.
Meredith, do you want to call on people for questions? And raise your hand, please, if you'd like to ask a question.
Sure. So there's no hands raised at the moment, but there's some interesting questions in the chat box. So I'll start out with one from Bernie Crespi, who asked, what would be the tissue function consequences, if any, of having so many non-cancer positively selected clones in the body? Would those clones function less well, contributing to senescence?
Yeah, that's a great question, Bernie. Good seeing you again. I would have normally thought that these would disrupt function, and I'm actually not sure that's the case. Certainly from the work that was done in the esophagus, they noted that the esophageal epithelium that had the clones was completely indistinguishable from that didn't have it. So at least structurally, it looked pretty normal. Functionally, that's a little harder to tell, but in fact, you might almost imagine that some of these mutations, like the NFK BIS genes, they actually noted those cells look better than not. In that case, ulcerative colitis is obviously just wreaking havoc on your gut epithelium. And so that's actually adaptive, not just in the sense that it provides an advantage to the cell, but it seems to actually improve the function of the colon. So you can imagine that might even be some, almost like a gene therapy strategy. But I would say it could probably go back both ways. I'm sure there are cases where we select for particular mutations as we age that do reduce function. And so it's probably a mixed bag.
So if I can just follow up, how commonly do you find those positively selected clones in germline variation among people? So if they are good for something, maybe they're just naturally present.
Yeah, I don't know if they're good for something normally. In other words, for most of the life of most people, probably that Notch mutation would not be advantageous. I don't know. I mean, when they've looked for cancer-associated mutations in the germline, clearly there's strong selection against them. I don't know if people have now gone back and looked at these, like the ones I was just talking about, the Notch1 mutants and the NFKB, the BIS one. I do know there are people that inherit Notch mutations, and honestly, I have to plead ignorance on that one. I don't know how that affects the tissue function. It's a good point.
Great, thank you. So it looks like next we have a question from Mary Woodall. Mary, you can unmute yourself and go ahead.
Hi, thank you. I was just interested to know if any of the genetic changes in the gut epithelium might be better thought of as a more multidimensional system where you've got the microorganisms that live there that might have say in selection and in their dialogue with the immune cells that also live there in terms of what constitutes benefit.
Yeah, no, that's a really good point, because it is a much more complex ecosystem. And certainly as we age, our microbiome changes. And it's been shown that the changes in the microbiome contribute to, increased gut permeability, which increases systemic inflammation, which then further feeds back to sort of reinforce this change in the microbiome. I'm less of an expert on ulcerative colitis, but I know that there's substantial dysbiosis going on there as well. And it's probably, it's certainly contributing to that inflammation. So in some ways, that might be another approach. So the question would be, is if you if you restored a more healthy microbiome, would you reverse that selection that's happening? Because clearly, in these individuals, we're seeing a lot of selection for those benign types of mutations. But I didn't show you down the list. There are some nasty, malignant type mutations also that can occur in those people, because people with ulcerative colitis have a substantially increased risk of colon cancer. So it would be really interesting to see. Probably you'd have to do mouse models to how changing the microbiome could influence those selective landscapes, those adaptive landscapes.
Yeah, I think that could be real interesting. Thank you.
Yeah, sure.
Great, thank you. So it looks like next we have a question from Charlie Nunn. Charlie, you can go ahead.
Hey, James, I have a couple questions, but I'll ask one first and see if there's time for a second one later. Some cancers are known as childhood cancers. Some leukemias, sarcoma, some sarcomas. I wonder if this perspective has anything to say about those cancers.
In fact, we actually did some computational modeling. So when we were doing this age-dependent computational modeling, it's even in the eLife paper. Without trying, we got the childhood leukemia bump. And because we are modeling a classic, what we knew about the hematopoietic stem cell pool and about how small it was at birth and how it grew. So it's clearly very small at birth, because it particularly, they estimate, dozens or, during fetal development, up to maybe a few hundred at birth. And when we did this modeling, we got clonal expansions that occurred early in life that mirrored the leukemia bump, that, increased risk from about 0 to 5 years of life. And when we went back and played around with the model, we showed that was due to basically drift. So because it's an expanding pool and because it's a small pool, you can get random expansions. And then we showed there was no need to infer a fitness advantage to the clone to get these expansions. So we kind of think what happens is that at birth, some individuals, just by chance, bad luck, basically, they have an expanded clone, both due to the bad luck of getting the mutation and the bad luck of drift-driven expansion. And most of those individuals, what happens is that clone's going to contract as the pool gets bigger. So basically stabilizing selection gets stronger and stronger. But in some fraction of those individuals, maybe they have another exposure early in life, or maybe they have something else that leads to sustained selection for that clone, and that can lead to a leukemia. And some of Mel Greaves' work really ties in here, because he's shown this associated with sort of delayed childhood infections and an increased risk of leukemia. And the idea being, in modern industrialized worlds, we keep our babies very clean, and then they suddenly go into a context like daycare at age 3 or, kindergarten or age 5 or 6, and they get sick a lot. And it's that sustained inflammation that might select for clones that are already present. So that's a bit hand wavy. The one thing I will always say is any exception to that late life rule tends to follow one of 3 things. One, it tends to be rare. So, whatever genetics is contributing to that is hard to select against. You can imagine the relative risk of a leukemia relative to death by infection was minuscule. And it tends to be something that's more modern, meaning it's more in the current age than an old age. So in other words, natural selection over, tens of thousands of years hasn't had time to take care of it. And the final one is they often tend to be associated with infections. So with pathogens that, have their own evolutionary agenda. And so I think a lot of exceptions to that late life pattern fit one of those 3 categories.
Great, thank you. Looks like next we have a question from Alicia DeLuise. Alicia, you can go ahead with your question.
Hi, I'm just wondering, I've seen some articles recently pushing away from the somatic mutation theory of cancer. And so I'm wondering how you feel this fits into that theory and perhaps eventually moving away from it.
Yeah, well, it's funny. People often accuse me of that. They say I'm like anti-mutationalist. And I'm like, no, I just think mutations is one part of the equation. So I think they have been overstressed. I mean, so, the bad luck model of cancer essentially puts all of, a lot of modeling that is done of cancer has literally assumed that every factor, whether it's extrinsic, intrinsic, or genetic, it has one parameter, and that's how many mutations you get. And so what we'd argue is that, yeah, you absolutely need a mutational and epimutational landscape upon which selection can act. So I'm not anti-mutation. Obviously, mutations provide the fodder upon which selection acts. So you certainly need them. There are some people that have argued, for example, Mina Bissell and some others in California have argued that there's that environment, basically is a normalizing influence, and that it's not so much the mutation, but that changes in the environment basically change the phenotype. So not really thinking from a selection angle, but essentially what could be benign within a normal environment becomes malignant within an abnormal environment. And I'd say there's also evidence for that as well, but I don't think there's mutual exclusivity. We definitely know that you can even take malignant cells. I mean, the classic examples from, Beatrice Mintz, many years ago, where she took F9 carcinoma cells. If she puts them under the skin of a mouse, they form massive, nasty carcinomas. But if she injected them into a blastocyst, they would actually integrate and become normal functioning components of many of the different tissues. So that was sort of a first indication that malignancy can be tamed by its environment, and that's independent of selection for or against it. So I'm sort of a believer that a lot of different people can be right. We just have to figure out how best to integrate these different ideas together.
Great, thank you for the perspective. So we had a question from Frederick Adler in the chat. Frederick asks, it sounds like the same forces, time, cell divisions, exposure, and genetics, are leading to the breakdown of tissues, but that the difference in perspective is that these are more like quantitative traits in building up predictably rather than depending on a few specific mutations. Is that a reasonable interpretation?
Maybe. I would think about it. You definitely, I mean, in some ways, we already know the factors of cancer. They are time. We get older, we're more, there are exposures. Smoking increases cancer rates. So in some ways, a theory basically has to explain what we already know. It's not really changing that those factors are important. And obviously our inherited genetics. I mean, there's not really much arguing with any of those. So what we're arguing is that it is true. It's in part through to degradation of tissue, but it's not simply a linear a type approach. So in other words, as we age from 0 to 80, it's not a continuous decline in our tissue health. And you can see this in a number of different metrics, but you really see it in disease incidence. Disease incidence is largely delayed, whether it's heart disease, cancer, kidney failure, to, after 50. And so that indicates that there is a larger investment in preventing disease through those, years of contribution to our genetic future. And so maybe for about half of a century, it's a relatively fat curve with some drop, but then that curve really drops precipitously. And I think that's a major difference, is that you're not getting sort of this continuous selection for oncogenic events throughout life, because we would argue that they're probably just as well impeded at year 20 as they are at year 30, maybe only a slight bit worse at year 40. But then there's a really increase in that selective pressure because the tissue changes in a nonlinear fashion as we get older. I don't know if that answered the question.
Yeah, no, I thought that was a really good response. I mean, I guess it seems like you're kind of arguing about a positive feedback, right? That as things break down faster. And I think somebody had mentioned this in the chat. You could also have an alternative where things accelerate just because you keep passing thresholds, right? There's a certain number of defenses in the system. And if you've lost half of them, no big deal. You lose 3/4, things go crazy. And that doesn't require a positive feedback. I mean, which do you think is more essential to this?
I don't think it necessarily has to be a positive feedback, but it's funny because we got together a couple of years ago with, I think I saw Michael Hochberg is on this call, Daniel Promislau, and some others. We had this meeting in Santa Fe, and we actually have a manuscript that is under review that's about resiliency, because I think resiliency is really key here, that any system can take a certain number of hits before it starts dropping off. And simply with redundancy, we know that we have lots of redundant mechanisms on our tissues. Hell, the fact that you can make so many knockout mice that don't have phenotypes tells you something, right? Or that heterozygous, losing one allele of most genes seems to have minimal effect. So, and I think there's, obviously there's even redundancy at the cellular level. We don't have one stem cell, we have large numbers of stem cells. So every time a stem cell accumulates a mutation, the easiest thing is that basically the body gets rid of it. And it's beautiful work from Drosophila and other systems of cell competition where a malperforming cell is eliminated. But I think eventually these systems, basically it would have taken a greater investment to make them go even longer. And unless there was selection for that, maybe if you had reduced predation, you could have lived longer and continued maintaining these tissues. So it's not like it's not possible, but I think those systems basically have their limit in terms of how many hits a system can take. There is positive feedback, I would say, within the best example is within the gut epithelium. And I think it's because, maintaining a tennis court sized, surface area in our gut takes a lot of energetic investment. And when that starts to break down and you get entry of bacterial products and maybe even bacteria and other organisms into our system, that inflammation then causes dysbiosis, which then further causes, gut breakdown. So there does seem to be some feedback loop, but clearly through most of our life, that is kept in check.
Great, thanks so much. Looks like next we have a question from Sebastian Van Nuytven. Sebastian, you can go ahead with your question.
Thank you very much for your presentation. So my question is, do you believe that we would also see the same accumulation with age for other kinds of mutations, such as copy number aberrations or structural variants? Because most of the papers now focus on single nucleotide variants, or are these effects just way too harsh to be countered?
It's an interesting point. There's some really nice studies that just came out on the bladder. And like many of these other papers, they show that, yes, there's some accumulation of like structural variants with age, but it's pretty minimal. And I think that they are too harsh. In other words, I think selection, the ability of our normal tissues to mediate the stabilizing selection is probably limited. I mean, I think there's definitely stabilizing selection. And like I said, work from Drosophila have shown that you can make a clone that's less fit and it can be eliminated from an epithelium. But I think there's limits. I think that if you have a clone that has a relatively modest effect on fitness, it can be maintained to some extent. But if you have a clone that has a structural variant, it tends to be too disruptive. I think it tends to be more effectively eliminated. So I don't think it's that the structural variants aren't happening through life. I think they're being effectively eliminated. And great examples of that is during fetal development. We know that there are lots of chromosomal, aneuploid events that happen during fetal development, but they're very effectively eliminated. Competition is fierce. Cell competition.
Great, thanks. So it looks like we had a question from Randy Nessie in the chat. Randy asks, any comments on how manipulating the cellular environment can inform treatment strategy?
Yeah, it's a great one. I mean, I think in some ways this speaks more to prevention. Because I show examples with anti-inflammatories, but it's not like I'd recommend that people start taking anti-inflammatories because, we evolved a nice inflammatory system for a good reason, is it keeps us from dying of infections. In fact, was a very large study in Europe, the CANTOS trial, where they gave people who had already had one cardiac event, they gave them anti-IL-1 antibodies for a couple of years. And indeed, it reduced the chance of the next cardiac event, but amazingly, it reduced lung cancer incidence threefold. So, clearly that's great evidence that reducing inflammation reduces cancer risk. The problem is they had no increase in overall, survival, because they had just as many people now die of sepsis that didn't die of lung cancer. So we have to be careful with these strategies. I think we need to learn to tune. We need to learn to start to bring the microenvironment back to a more normal state, but I don't think we could take sledgehammer approaches. So I think the microbiome may be a great entry point. I think if we can learn to better maintain a healthy microbiome, I think we're going to go a long way to better maintaining tissue health.
Great, thank you. Looks like we have a question from Scott Kennedy. Scott, you can go ahead with your question.
Yeah, so good seeing you again.
Yeah, good seeing you.
So what about, you're talking about like IL-3. So what about other interventions like caloric restriction or rapamycin or things that seem to extend lifespan and even like modify cancer incidence, like caloric restriction in monkeys, 50% less,
Right. Yeah, there's always pros and cons. I worry about caloric restriction because most of those studies have been done in, sterile housing, with clean food. And so caloric restriction has been shown to reduce immune function. It's been shown to reduce wound healing. So those may not be things that the animals are going to deal with much in their cages, but in real life, it could be a bad thing. Not to mention that I'm not sure, let's say that my life was going to be 10 to 15% longer, but 30% more miserable because I'm always counting calories. You know, it's a bit of a trade-off. So, but I do think they link in. I mean, clearly caloric restriction stimulates the same pathways, like it inhibits the IGF pathway. It boosts autophagy. I actually think autophagy is going to turn out to be a real key in terms of investment strategies. They've even made mice that have boosted autophagy. They live longer, they get less cancer. And what was interesting is in their methods, they noted that the mice ate more to maintain the same weight. And they made nothing of this. It was just barely mentioned the methods. And I thought, wow, that's huge. Because this says why you wouldn't basically select for a mouse in the wild that has boosted autophagy. Because first off, the chance that it's going to make it to old age is about nil anyway. And they would have had to find even more food than they already had trouble finding in the first place to maintain their tissues. So a lot of these interventions, even things like exercise, they often tie into boosting autophagy, which boosts the somatic maintenance and maintains that landscape. So my strategy is screw caloric restriction. I'm just going to exercise more.
Great strategy. So we have time for probably a couple more questions. So if you have a burning question, feel free to raise your hand now. Next, we have a question from Abin Abraham. Abin, you can go ahead.
Hi, I was wondering how you think about tissues that have different rates of cancer. So for example, cardiac cells,
Right.
They're working for our entire life. So pretty demanding job, but we don't see a high rate of cancer in these cells. So where does the somatic mutation play in there?
Yeah, no, well, I think most somatic mutations are going to happen during cell divisions, during tissue renewal. So largely post-mitotic tissues aren't going to be subjected to that. And, honestly, I know that they've looked at mutation accumulation within like neurons, and you can find mutation accumulation, but I think the odds that you're going to get something that leads to a clonal expansion is going to be much lower. So most cancers are occurring in tissues that have some level of self-renewal, and sometimes that could be stimulated by, our exposures. So even though the liver turns over at a relatively low rate, if somebody drinks a lot or if they have hepatitis, there's a substantially higher turnover rate. So not only are you going to increase the odds that you get a mutation, but you're also going to now have a perturbed environment for that tissue. So yeah, I don't think that fully addresses the variance between tissues, but certainly we do notice a lot of cancers arise in tissues that turn over at a reasonable rate through life.
Great, thank you. So we had a question from Sonia Cavagelli early in your presentation. Sonia asked whether there were sex differences in the incidence rate curves versus age.
Oh, I love it. That's a great question. And does anyone know the answer to that? Well, first off, who lives longer on average, men or women? Everyone knows that one, right? Yeah, I saw Alicia raise her hand. Yes, women. Women live longer, right? So women live longer. And so essentially the curve of physiological decline, we feel sort of mirrors the curve of death. Probably, obviously, the physiological decline is a little left shifted to it, but men decay earlier. And it's sure enough, if you look at cancers, so what we did is there's actually a figure in the book where I kind of modeled it, but we looked at cancers on SEER that weren't sex dependent. So obviously we rolled out breast cancer and prostate cancer, and things like AML are actually indeed leftward shifted for men. They come up earlier. And so there does appear to be some truth for that. I don't think people have looked in a systematic manner, but the few cancers we looked at that were largely aging dependent, in other words, they didn't have an extrinsic factor like smoking, do indeed show that shift in the curve. So I think that is a very relevant factor. And you might ask the question, well, why do men age faster? Because there's always some man in the audience that asks, well, why, I can keep reproducing till I'm 70. And I'll always respond, well, not if you're dead. And so, men actually, males in many like vertebrate species actually have shorter lifespans because they basically have higher extrinsic mortality throughout life. And even in modern societies, a 20-year-old to 30-year-old male is far more likely to die from an accident than a female. You know, I always like to say, how often have you heard of a woman dying or a girl dying after saying, hey, y'all, watch this? It's a very male thing to take chances. And it's basically because men invested even in things like fighting and robustness. They didn't invest in the long game because basically it was either breed early or forget about it. And that may be an exaggeration, and Randy's probably going to kill me, but that's at least my simplistic interpretation of it.
Sonia, did you have something to add to that? I saw that you had unmuted yourself.
Yeah, I was thinking just as a follow-up that you had referenced this in when you were talking about those incidence curves, that right, men can still reproduce, you know, forever, whereas women are cut off. And so would natural selection work in a different way on females that have a kind of a shorter reproductive lifespan?
Yeah, no, there's a lot of interesting thoughts on that. I mean, but I think that, you know, you would only have selection for continued maintenance of the male soma if it was beneficial. In other words, if that variant that like did greater investment was more likely to, you know, on average provide more offspring that survived. And I think if the men are dying at a more rapid clip because of fighting or competition for mates and so forth, or stupid activities, that just less likely to have that pressure. And of course, there's a whole field on understanding, you know, why do women undergo menopause? And that in itself is a very interesting question, you know, in terms of what are the selective pressures for menopause. Obviously, there's the grandparent hypothesis, and there's the idea that you know, women, you know, of the human species have a reasonably high rate of death at birth, you know, during childbirth for at least most of our evolutionary history. And so every time a new baby's born, that woman is essentially rolling the dice. And if she keeps rolling the dice, it becomes a greater probability that she's going to die in childbirth, and thus her previous children and haven't reached some critical age might not survive. And so, and actually, Fred Thomas and some of their colleagues have also come up with other ideas of there might be tumor suppressive functions for having menopause.
Right, cool, thank you.
Sure.
Great, so next we have a question from Ashley Painter. Ashley, you can go ahead.
Hi, I'm Ashley. I'm Daniel Promislow's grad student. And I study, I guess I should.
No, in fact, he's talked about you. We're talking about you.
Start my video, hi. And I also work, I work with Scott Kennedy also.
Yeah.
But so when I think of cancer in different species as well. And so I was wondering how we might start to understand why some species have evolved better mechanisms against cancer. Is this based on size alone? I mean, we know that naked mole rats have defenses against cancer. We know that elephants have like gene duplicates of TP53. So even opening it up more to like other species, why is it that humans get so much cancer? How can we better understand that?
Yeah, I would say that I'm not sure there's as much of a differential as there's been made to be, because I would say most animals, and actually Rob Noble and Michael Hochberg did some estimations of this. And essentially, I believe they estimated that for animals in the wild and in the absence of human interventions, most animals, probably no more than 5% will suffer from cancer. So it's true, you know, people often refer to mice. Well, mice, you know, half of them die of cancer. Well, that's mice in our labs. In the wild, I'd say very few mice die of cancer. So in some ways, mice may be just as good as tumor suppression as a naked mole rat. And it's because basically the mouse strategy is to live for a few months, reproduce like crazy, because you're not going to be around very long. And it's true, elephants, you know, can go through 100 years, but I think essentially that's all tuning of those maintenance programs. And so, you know, if an elephant, because an elephant, you know, except for recent times with humans, they could largely avoid extrinsic hazards. And so that meant that they could invest in the long game, just like humans could, because of some of the mechanisms that we evolved to avoid extrinsic hazards. Other animals like turtles, even birds have, so it's not simply related to size. Birds have lower cancer incidence and have longer lifespans because by flight, they avoid extrinsic hazards. So they could basically extend out that maintenance. And the longer they extend out the maintenance, the less cancer is likely to develop during that period, at least according to our theory. I think, you know, there's more than that. Obviously, that same investment in maintenance is going to invest in immune function and other things that could also function to reduce cancer incidence. So I think that the naked mole rat one thing we often find is that when we bring animals into captivity, if the animal's already long-lived, we don't tend to do much better in captivity. In other words, we don't tend to extend their lifespan much. But if an animal is short-lived, like a mouse, we can massively extend their lifespan, allowing us to see essentially what happens when you keep an animal around for much longer than it'd be around in the wild. And then I think we see all these disease incidence curves. So I'm not totally convinced that the naked mole rat is that incredibly special. I think it definitely has tumor suppressive mechanisms. All animals do. But anyway, that's my explanation. Any other, does that answer your question, Ashley?
I don't know.
All right. So it looks like we have one final question from Joseph Graves. Joe, you can go ahead with your question.
Yeah, I'm really sorry I missed the beginning of this talk because obviously it was really interesting. But one of the points I always like to make is this claim about the potential of males to have reproduction for a very long time is dramatically overstated. Just because there's a potential that it could happen, is the question of, does it actually happen?
Yeah.
And certainly in any species where sexual selection plays a role, the capacity of older males to compete for female attention is going to drop dramatically.
Yeah.
And that's in a lot of species. So I think people need to get off that bandwagon.
I totally agree with you. Yeah, it's, you know, when you're reproduction, when you're older, when you're frailer, I mean, because given that a lot of times it was competition for mates, you would have been, you know, not been very competitive, and particularly if you're dead. Death puts a real damper on reproduction.
Yeah, I agree. Can't have sex if you're dead.
Okay, I think we're going to close out on that great comment. Thank you for that, Joe. And thanks, James, for a fantastic presentation. This is really fascinating. And for all the really great questions. This was a really nice discussion. Thank you, Meredith, for helping to organize. And before we totally close out, I want to share my screen and show you some of the upcoming Club EvMeds. Hopefully you're seeing my screen now. You can see our website, the talk we just heard. And next week on Tuesday at 1 o'clock Eastern time, we're going to be hearing from Tobias Lenz, who's going to be talking about pathogen selection for HLA-MHC gene diversity and its consequences for autoimmunity and cancer immunotherapy. After that, we'll be filling these in soon. We have Dan Blumstein, Sylvia Kramer, Phil Starks, and Noah Rosenberg. So a great lineup coming. Please stay tuned for these talks, and we hope to see you at future Club EvMeds. We also have a postdoc presentation session where 3 postdocs will be presenting their work as well. So thank you, everyone. Thanks for participating, and we hope to see you again next week. Thanks.
Thanks, everyone.
Thanks, James.